Friends, seeking the blessings of the Almighty. I now call upon the executive member of the Newberg crew, Ms. B. J. Lakshmi, for the invocation.
Satyavratham Satyaparam tu Satyam Satyasya Yonam Nihitancha Satye Satyasya Satyam Vrita Satya Netram Satyatmakam Swam Sharanam Prapatnaha Vani Gunanukathane Shravanau Kathayam Attaucha Karmasu Vanastava Padayornaha Smityam Shirastava Nivasa Jagadranam Ististatham Darshanetu Bhavathanunam Namo Bhagavate Tasmai Krishnaya Adbhuta Rupanama Vidhetena Jagat Kridatiyo Yataha.
Light is a pretty universal symbol of truth, knowledge, and understanding. The lamp shows everyone how to rise upwards and dispel darkness. To symbolize the inauguration of this international conference, we will now have the virtual lighting of Kuthuvelakku. I request the esteemed speakers and chairpersons of this conference to kindly switch on your video for this traditional event. Please, sirs and madams.
Dr. Ravichandran, can you please switch on your video? Principal sir, video கொஞ்சம் ஆன் பண்றீங்களா? Lighting of Kuthuvelakku. Sir. Principal sir.
Hello?
Sir, video கொஞ்சம் ஆன் பண்ணுங்க சார்.
ஓகேவா?
Yes sir.
The greatest leader is not necessarily the one who does greatest things. He is the one that gets the people to do greatest things, said Ronald Reagan. We have a great leader, the one who created the spark for this conference, the co-convener of this conference, and the head of the Department of Biochemistry, Dr. P.T. Srinivasan. I now invite him to welcome this august gathering. Sir, please.
Jai Sri Krishna. Good morning to one and all present in this conference. Respected Secretary Sri Ashok Kumar Mundraji, our beloved Principal Captain Dr. S. Santosh Babu Sir, Honorable Chief Guest Dr. Manikam Ravichandran, Dean of Faculty of Applied Sciences and former Vice Chancellor of IMST University, Malaysia, and revered keynote speaker Dr. Vasan Sambandamurthy, CEO of India Alliance Hyderabad. And we have ardent plenary lecture speakers, Professor Selvara Subramaniam, Head Statistical Analysis and Data Management Solutions of UK, Dr. Anuja Matthew, Associate Professor of Research at Department of Cell and Molecular Biology from University of Rhode Island, US, and Dr. Kavita Rajshekhar, scientist, ICMR, Department of Health Research, Ministry of Health and Family Welfare and Government of India. Distinguished chairpersons for the paper and poster presentations, Dr. Neetu Nigam, Dr. Sujata, Dr. Vijaya Srinivasan, esteemed delegates from national and international level, heads of various departments, faculty members, sponsors, media persons, technical experts, my dear research scholars, students, and other online viewers present in this conference. This conference is a noble endeavor of Knowledge Summit in general. So it's a moment of extreme pleasure to welcome you all to this two-day international conference on infectious disease in an era of vaccines, organized by my department, Biochemistry, Dwark Das, Govardhan Das, Vaishnav College in Chennai. So since the inception of this college in 1964, the college is imparting a value-based education to enthusiastic learners in diverse disciplines in various fields of education, including arts, commerce, science, technology, and management. So with immense pride, I admit that the Department of Biochemistry of this college, which has started in the year 1995, has the state-of-art facilities, and it has grown to leaps and bounds in the period of 25 years. The vision of the department is to bridge the gap between the academia and the industry, and striving to be the center for excellence in biochemistry. I'm extremely glad to register here that the department has completed 25 years of its jubilant journey, and this conference is organized as a mark of the Silver Jubilee celebration. On this note, I extend my hearty welcome to our Secretary, Sri Ashok Kumar Mundraji, the management to this conference. But for their support and guidance, we would not have made this journey so far. I'm also privileged to welcome our beloved principal, Dr. S. Santosh Babu, who has taken this conference to the state that it has seen the reality of the day today. Thank you, sir, for being here with us. You are the motivating factor and the guiding spirit of all our endeavors in our department. I take the pride in welcoming you to this international conference and to commemorate the Silver Jubilee of our department. Now, I especially welcome the versatile, dynamic, and intellectual chief guest of the day, Dr. Manikam Ravichandran from Malaysia, AIMS University, Malaysia. Sir has a very strong background in medical microbiology and has been awarded many national and international awards and products, and he has been given patents in various products. I also take immense pleasure in welcoming Dr. Vasan Samandamurthy, CEO of India Alliance and Hyderabad. And I take immense pleasure in inviting the plenary speakers, Professor Selvaraj Subramaniam and Dr. Anuja Matthew, Dr. Kavita Rajshekhar from the various respective disciplines. So we have around more than 300 registered participants for this conference, and we have received many posters and paper presentations. So I take immense pleasure in inviting you all to this wonderful two-day international conference, which I am sure will serve as a portal to the exchange of knowledge of various aspects of this vaccines, infection, and in particular about the COVID-19 pandemic that is prevalent today. So since this international conference is built on this context and with the intent of active and interactive learning, I hope that all of you will have two most productive days of interesting and stimulating discussions. I sincerely wish that the conference will be a great success. I once again extend my very warm welcome to everybody present here. Thank you for giving me this opportunity here. Thank you. Over to the organizers.
Thank you, sir. Great teachers and principals can help to close persistent achievement gaps, improve student attitudes, and build habits of mind that can change a student's life. Our beloved principal emulates these qualities perfectly, and it is a pleasure to have him amidst us. On behalf of the Department of Biochemistry, I now invite our revered principal, Dr. S. Santosh Babu, Member Syndicate, University of Madras, to deliver the presidential address. Sir, please.
Warm good morning. I welcome all the dignitaries on the dais and off the dais. First, I like to welcome our chief guest for this inaugural function, Dr. M. Ravichandran, Dean from Amherst University, and keynote speaker, Dr. Vasan Samandamurthy from DPT Welcome Alliance, Hyderabad. And also, I like to congratulate the PG Department of Biochemistry, Research Department of Biochemistry, which is conducting an international conference today in the silver jubilee year of the department. The faculty members and the students have shown keen interest in conducting this program successfully. Though we caught in between a pandemic, the department was continuously engaging themselves in all activities like this. And based on that only, today we have this international conference. I would like to make use of this opportunity to congratulate all the beneficiaries who are attending this today's program. And also I like to quote that the department have hosted more than 25 webinars in the 25th year, and through which the students have benefited, where they are able to gain the knowledge on scientific data and scientific knowledge. Many of the department alumni of this institution have contributed for this reputed institution. They also have served as a resource persons for various webinars. This, I am sure, must be very inspiring for the current students. And also, in the present higher education scenario, the role of faculty and the role of students have changed. Apart from the regular teaching learning process, it is the duty of every faculty to attend the program successfully, and also they used to have a warm greetings to the people. So this, in the COVID period, we are able to conduct this international conference on behalf of Dwarakadas Govindas Vaishnav College. So in this time, our secretary also joined today. And my best wishes for all of you. And also my secretary says a few words to you.
Good morning to one and all.
Good morning, sir.
I wish this seminar a very grand success, a very unique seminar. This time we are having an international seminar in this COVID time, and I hope that everyone will enjoy and benefit from this seminar. Thank you.
Thank you very much. Thank you, sir.
sir.
Once again, I like to welcome the chief guest for today's program. Thank you.
Thank you, sir.
Thank you, sir. We thank our Honorable Secretary and the Dynamic Principal for the guidance and support and blessings that are extended always. Thank you, sir. The intellectual mind is always filled with questions: where, when, how, why, and the like. I now invite Dr. Sangeeta Satyanarayan, Assistant Professor, Department of Biochemistry, and the Organizing Secretary of this conference to present the dynamics, the when, how, and why now for all of you. Ma'am, please.
A very good morning. The 21st century has witnessed a wave of severe infectious disease outbreaks, which has had a devastating impact on lives and livelihoods around the globe. The 2003 severe acute respiratory syndrome coronavirus outbreak, the 2009 swine flu pandemic, the 2012 Middle East respiratory syndrome coronavirus again, 2013 to 16 Ebola virus epidemic, 2015 Zika virus pandemic. All of these resulted in substantial morbidity and mortality by spreading across borders to infect people in multiple countries. As I'm sure that none of us were born in 1918 during the flu pandemic, the world has once again witnessed a pandemic of epic proportions in the current century. But one thing has changed tremendously. The advancement in science and technology has prepared us to face the current challenge in a better manner. Against the tide of losing lives and calling this a new normal, science and technology continues to remain a silver lining during this pandemic. Against all the odds, the unassuming scientists around the world were working on developing different technologies as therapies for many diseases. Little did we realize that all those years of research has led us to develop a vaccine in such a short time. The swift development of the severe acute respiratory syndrome coronavirus 2 vaccine speaks to the efficacy of modern science in rapidly countering threats from emerging pathogens. The COVID-19 pandemic took the world by surprise. While the first documented case was in December of 2019, it took just 3 months before the World Health Organization would declare the COVID-19 as a pandemic. There has been substantial amount of uncertainty surrounding this condition. How long will it last? Is it the true mortality rate that is reported? Will there be lingering health and economic effects? When faced with this level of uncertainty, it is tempting to look to history for guidance. Among the set of historical pandemics and epidemics witnessed by the world, 1918 influenza pandemic is appealing for two reasons. First, the 1918 pandemic is the most recent historical event to match or even exceed the scale of pandemic compared to 19. Second, 1918 pandemic occurred after a series of improvements in data collection and preservation. Because of these similarities, the 18 pandemic offers a unique opportunity for comprehensive and systematic analysis across a wide range of countries. Though COVID-19 has many important parallels with 1918, there are also some important differences. One key difference between the two being the quality of life and overall life expectancy were much lower in 1918 than today. Cities in 19th and early 20th century were polluted and the risk of contracting and dying from an infectious disease was high. Throughout the 20th century, a series of innovations brought about dramatic improvements in health and longevity. Cities discovered and invested in new technologies to eliminate waterborne diseases like cholera and typhoid fever. Sulfa drugs were discovered in 1930, providing the first effective treatment for infectious diseases. And then on, there has been no looking back. The theme for the conference evolved, keeping in mind all the above, and also from what we are going through, that is living in the new normal. And the theme can never come at a more appropriate time, as this week, October 17 to 23rd, also coincides with the International Infection Prevention Week. The two-day conference offers 5 plenary lectures from scientists of eminence from various fields, definitely making the conference an enlightening one. Our first plenary lecture is by Dr. M. Ravichandran, Dean of AIMST University, Malaysia, whose research interests lies in the field of molecular diagnostics. His contributions to the field is voluminous. Dr. Vasan Sambandamurthy is yet another stalwart from DBT Wellcome Trust Alliance, Hyderabad, with over 2 decades of experience in vaccine discovery and drug development. Recording, analyzing, and interpreting scientific data is always important and particularly more relevant during a pandemic. Professor Selvaraj Subramaniam from UK specializes in statistical analysis and data management solutions. Dr. Anuja Matthew from the US is a cell and molecular biologist with a rare blend of teaching, research, and industrial experience. Our last speaker, Dr. Kavita Rajshekhar, is a senior scientist of ICMR. She has a very progressive professional career. She starts with teaching and then moves on to research, and now is working hand in hand with the Government of India to ensure that policy decisions made by the Ministry of Health and Family Welfare are implemented as advocated. Hence, the conference offers ample opportunity for scientific deliberation on key areas of research. Paper presentations on topics like artificial intelligence in health, role of bioinformatics in target identification, phytotherapy, secondary metabolites as drugs, nanotechnology in infectious diseases, antibiotic resistant microbes, and so on. I thank all the resource persons for making this possible, and I congratulate the delegates for making use of this opportunity. Wishing this endeavor all the success that it rightly deserves. Thank you.
Thank you, ma'am, for a lucid portrayal of the dynamics of this conference. Now we have come to the much awaited moment of the inaugural session. We have an eminent academician, Dr. Manikam Ravichandran, to inaugurate a conference. I invite Professor K. Nagalakshmi of the Department of Biochemistry to introduce our esteemed guest. Ma'am, please.
A very good morning to one and all present here. On behalf of the Department of Biochemistry, I take an immense pleasure in introducing the chief guest of the session, Professor M. Ravichandran, Dean of Faculty of Applied Sciences, AIMS University. He obtained his MSc Medical Microbiology from Christian Medical College, Vellore, and gained his PhD in Biotechnology degree from Anna University, Chennai, India. Currently, his research interests are genetically engineered cholera vaccine and formulation, molecular diagnostics and phage therapy. He has 70 publications in international journals, supervised 40 postgraduate students, filed 8 patents, and commercialized 3 products on diagnostics. He has been awarded 44 national and international awards for the academic and research excellence. He was an associate member and committee member on Top Research Scientists Malaysia, TRSM, of the Academy of Sciences of Malaysia, that is ASM. He was the former Vice Chancellor of AIMS University, Malaysia. Sir, it's a deemed privilege to invite you for the chief guest lecture. Please welcome, sir.
Okay, thank you, Dr. Nagalakshmi, and good day to everyone. I think a lot of people from different time zones are here, and thank you for inviting me to the two-day international conference on infectious diseases in the era of vaccines, organized by Dwarakadas Govardhan Das Vaishnava College, Chennai. Vanakkam to everyone. Greetings from AIMS University. And I'd like to give my sincere appreciation to Sri Ashok Kumar Mudra, Secretary Dwarakadas Govardhan Das Vaishnava College, and the respected Principal Dr. S. Santosh Babu, and Head of Department of Biochemistry, Dr. P.T. Srinivasan, who has been championing and been contacting me to give this talk. And my sincere thanks goes to Prof. Sangeeta Satyanarayanan and Department of Biochemistry, and who has been in touch with me throughout this event. I've been impressed by the arrangement made by the conference committee. All the hardworking committee members, dear students and staff members of DGVC, and participants and other speakers, doctors, all the speakers today, Drs. Vasan, Selvaraj, Anuja, Alan, Edelman, and Kavita. And it's indeed a pleasure to join this event. And good day to everyone. So I understand that Department of Biochemistry has played a vital role in organizing this event as a part of the Silver Jubilee celebration. And I was asking about the alumni data to Dr. Srinivasan. He was telling me that has produced more than 1,250 quality undergraduate and postgraduate students. And I'm impressed by that. And they have been placed in reputed places like Biocon, Abbott, and Sankara Nethralaya, NIMHANS, and CCMB. And he was telling a lot of reputed universities and places there. Some of them have started their own startups, and very delighted to see the fruition of the alumni. I'm very impressed by the service rendered by Dwarakadas Govardhan Das Vaishnava College, a linguistic minority institution established since 1964 with the sole purpose of imparting knowledge. In this current commercial world, it's very difficult to see such a value-based educational institution. And it has been a heaven for all the enthusiastic learners and the needy people. And The Department of Biochemistry has been offering undergraduate and postgraduate program, bridging the gap between the industries and academia, and doing a very good service. Not but least, but I would like to congratulate the Newburgh crew, the department club who has organized this particular event. In Malay, there's a word called Tanya, means congratulations to all who have organized this event. Let's not talk too much about COVID, but that we have to thank COVID that we have been remodeled since I would say December 2019 or March. The WHO declared 11th of March 2020 as a pandemic. And we have to congratulate ourselves that we have survived this pandemic situation, and we have successfully learned too many things, including this Zoom thing, which we never imagined how we are going to use it. And we have to thank all the frontliners, doctors, and all the people who are in the nurses and everyone, each and everyone who helped us to bring this to the situation. I would like to thank one group of hidden frontliners. They are biologists, the biotechnologists, the biochemists who developed the vaccine, who developed the drug, who developed the diagnostics. Now we can do the testing, and we are able to live in this endemic phase of this pandemic. So I'd like to congratulate all the biotechnologists and biologists or biochemists who are here for putting their contribution to coming out of this COVID. I just want to give, since it's an infectious disease conference, I just want to, I don't want to talk more about disease. We will be giving our lectures on this area, and all the speakers are lined up here. So I don't want to give another infectious disease lecture over here. So the time given, I thought I'd give some of the things which is going in my mind. And there's always a race towards, we call publish or perish, publish for publish, or some people say publication for the own sake. There is a tremendous pressure on the academicians worldwide to publish, like ranking, QS World Ranking, Time World Ranking, and everyone going towards that that kind of publication. And the parody is that many of the publications are not being cited, and but they publish in an index journal and so on. And we can see a good example in this pandemic, how many research has helped to come out of this pandemic. Okay, we can say that we have millions of researchers there. How many researchers really resulted in quality publication that has impacted the society? So that's very important. I think the rule of the day should be research work should have impact on the society. And like this organization has been developed to impact the needy students, the research also should impact the society, uplift the poor, and uplift the bottom billion. There are a lot of people, billions of people in the world who live with $1 a day. So we have to make sure that our research brings benefit to the society. Okay, publication alone might not be a good matrices to evaluate. And I have a small message for the students. So there is an ocean of knowledge there. There's something called MOOC, Massive Open Online Courses. In this pandemic, I think you all have got used to it. Many are primarily offered free. And that you can do courses like CGMP, good manufacturing practice, good laboratory practice. So some of the regulatory requirement, you can get well-paid job as in CROs, clinical research organization. This is a small example I'm giving you. Spend your time usefully and registered for those courses, and you'll be able to add value to your CV. Important thing in the current day is networking. Network with your peers, seniors, and definitely they'll be able to guide you. Okay, networking is very important. And you can go for a startup. I'm very impressed to know that you have many startups coming from this particular college. and you should be able to do your startup and become an employer. So in the COVID era, there are a lot of competition happening online. You don't have to travel to US, you don't have to travel to UK. You can spend money on flight ticket. You can join competition, search for international competition, join quiz competition, debate competition, conferences. You can join online and win prizes. So I don't know, this pandemic is going to end. This is the last call. I think you can try to upgrade your CV by joining all these conferences. The most important thing about Indians are very, I don't want to be very nationalistic and say that we are very bright, but most important thing, you can see a lot of CEOs internationally, we can see the Indians are helming that position. And I did so here in Malaysia. Character, credibility, soft skills are the need of the day. Knowledge is out there. You can learn anything very easily, very fast. Okay, it's not a difficult thing. Knowledge it's out there. So when we have such a August event, how you can communicate, the soft skills, how, let's say you come, credibility, you say you come to that place at that particular time, you start a meeting on time. I'm very impressed by this conference started, and we don't even have to use the word Indian Standard Time. So these are the most important thing, and you have to have be one of them. Be helpful to people and planet, environment, people, animals, everything. You can, if you see biotechnology, biotechnologically, every cell has a genome, and genome, we all are relatives. Okay, we are 98% related to monkeys and chimpanzees, and we are almost 99.9% similar to each other. Even trees share our genes. Sorry, we have to say we share genes with the trees. So every living organism in this planet are our relatives. We all originated from the same origin. So we should be grateful and share the planet, and inclusivity should be there. We have to make sure that we all live together. Essentially, intelligence and character is the true goal of proper education. Educating the mind without educating the heart is no education at all. This was said by Aristotle. So I wish a grand success for the 2-day international conference on infectious diseases, an era of vaccine. So hearty congratulations to the Department of Biochemistry and Dwarakadas Govindan Das Vaishnava College. And I'm very happy to inaugurate this conference. Thank you very much for inviting me. I wish you all good luck. God bless. Thank you.
Thank you, sir, for your warm words of appreciation. Sir, we'd like to register that our students have undergone a lot of MOOC courses and are still registering. And in fact, the good laboratory practices have been included in our curriculum of late.
Excellent.
And I'm sure we'll also make sure that we do a lot of research for the benefit of the society, as you have suggested. Thank you, sir. A conference is a platform for sharing knowledge on recent advancements in various fields. True to this, we have received a number of abstracts for paper and poster presentations. All the scientific content has been documented in our conference proceedings with ISBN number. Since this is an online conference, now we'll have the video of the release of the proceedings of this conference by our revered principal, Dr. S. Santosh Babu. The first copy was received by our beloved HOD, Dr. P.T. Srinivasan. There's a slight technical lag. Participants, please stay tuned. Participants, kindly mute your mic. We are sorry for the inconvenience. Please stay tuned. Nothing great will be achieved without great men, and men are only great if they are determined to do so. We have a great personality, Dr. Vasan Samandamurthy, amidst us to deliver the keynote address. I now invite Dr. G. Sriram Prasad, Assistant Professor of the Department of Biochemistry, to introduce our guest. Sir, please. Sir, kindly unmute your mic.
A very good morning to all. It gives me immense pleasure in inviting Dr. Vasan Samandamurthy, Chief Executive Officer, India Alliance, Hyderabad. Dr. Vasan joined the DBT Welcome Trust India Alliance with over 20 years of professional experience in vaccine discovery and drug development. Dr. Vasan completed his postgraduate degree in medical microbiology from Christine Medical College, Vellore. He gained his PhD from Tuberculosis Research Center in Chennai and completed his postdoctoral training as a Howard Hughes Medical Institute Fellow at Albert Einstein College of Medicine in New York. He has previously worked for major global corporations like Novartis, AstraZeneca, and Biocon. At these organizations, Vasan has held senior leadership positions, provided strategic direction, and orchestrated major business operations to develop affordable medicines across multiple therapeutic areas, including oncology, diabetes mellitus, infectious disease, especially in tuberculosis and malaria and inflammation. He went on to leverage his academic training and real-world experience in managing large research and development portfolios, interdisciplinary and cross-functional global teams, regulatory submission, and global alliance to advance diverse classes of molecules into clinical development. In spare time, Vasan is a prolific long-distance runner, avid trekker, and classical music enthusiast. We are eagerly waiting for your lecture on power of industry, academic collaboration, discovery, and development of a new novel anti-malarial molecule. We welcome you, sir, to deliver the keynote address. Thank you, sir. We welcome you, sir.
Thank you so much, Dr. Sriram, for that wonderful introduction. Good morning, good afternoon for friends who are across the world. It really gives me great pleasure and privilege to deliver this keynote lecture. At the outset, I'd like to thank the organizing committee for giving an opportunity to be a part of this 25th year celebration of the Dwarka Das Govardhan Das Vaishnav College. And it's also a tremendous opportunity for me to reconnect with some of my college mates and seniors who we studied together at CMC Vellore. So at the outset, I would like to special thanks to Sri Ashok Kumar Mundra, the Chief Patron of the college, as well as Dr. Santosh Babu, the principal. Special thanks to my friend, Professor P.T. Srinivas, Dr. Sangeeta, the host for today, Dr. Maria Carolyn. So, and a warm welcome to the faculty, the students, the Newburgh Club, which really made this opportunity to bring all of us together into a platform to really exchange our knowledge, ideas, and have an engaging conversation. So I have spent more than 20 years working on infectious disease, something very close to my heart. I trained microbiologist by background. So I've got trained in vaccines, then spent almost 2 decades in the industry trying to develop new drugs for tuberculosis, which is a major killer compared to COVID, and then also worked on malaria. So what I intend to do today is basically to walk you through a journey of how important it is for industry and academia to come together to really converge their excellence, converge their knowledge, and the know-how to really benefit the society. One such example is what I'm going to share. Please let me know if you can see my, if the organizer, could you please enable a screen sharing, please? I get a message which says host disabled. I still cannot share my screen. And while I wait, a special thanks to our chief guest today, Dr. Ravichandran, who was my senior in college. Great to see you, Ravi. And my batchmate, Selvaraj, who is dialing in from Scotland. Dr. Maria, could you please enable screen sharing?
Yes, sir, we are doing it, sir.
Sure.
Please.
This is a new norm, right? We all live in a virtual world. We have to be patient and ensure that the platforms work. And definitely we have seen a huge leap in the technological advances that have happened in the last 1.5 years since the pandemic. Okay, let me just see. Yes. please let me know if my screen is visible.
Yes, sir. Yes, sir.
Excellent. Once again, good morning. Kale Vanakkam to everyone. So I'm not going to talk too much about the pandemic, all of us have heard, but here is a story of hope. And here is a story of how the best minds can come together and really make a remarkable difference in terms of developing new medicines, new vaccines, and how we can impact the society. So I'd like to start with this famous quote by Dr. Albert Einstein. It says, "In the midst of every crisis lies great opportunity." I'm sure all of you agree with me that how the human race really collaborated, worked together, and really came out successful, because we did lose a lot of life. But definitely the heart of where we are today is because of science and technology. So science, R&D has taken the center stage in what we are looking at. So what I intend to do now is to give you an example, illustrate an example, a real life example, which happened 8 years back. This is a disease which has been a huge burden to the developing world, India, Africa, and a lot of Southeast Asia. And what I intend to do in the next 40 minutes, 35 to 40 minutes, is to show you how the best minds in academia, industry, private organizations, philanthropic organizations, as well as government agencies can come together to really converge on making a huge difference to the humankind. So am I audible and my screen is visible? Just want to make sure that.
Yes,
Yes, sir. It's fine,
we can
sir.
see your screen.
Great. Yeah. So what is shown here is how do you increase the impact of industry academia collaboration? You are all coming from academics, which is the bedrock for education, getting the best minds trained with ideas, coming with a lot of depth in fundamental research. Industry, on the contrary, really applies the knowledge that is developed in academia in terms of world-class processes, mechanisms, in terms of how do we make decisions and on how do we move things forward with the sole purpose of developing a product. The product could be a diagnostic, which you will hear subsequently from some of the speakers. It could be vaccines, you'll hear from a chief guest later this morning, or it could be new drugs, which I'm going to talk about. So as long as there is a common problem that is identified, which brings value to the society from the academic partner as well as with the company, and then the collaborative partners look for a source of funding and resources. And then that kickstarts a collaborative framework, which really helps to develop new knowledge. And this new knowledge benefits the researcher in terms of publications, in terms of patents, and it also benefits the company in terms of improvement, process improvement, or developing a product. And finally, the society gets the value of this, the partnership between academia and industry. And this whole industry academia collaboration is built on trust and relationship. So let's spend a minute on what are the enablers for collaboration. As all of us know, academicians are very keen on publication because that is one of the milestones that is used to calibrate the success of a researcher in academic labs. I did that 15 years back as a postdoc researcher. Whereas for the industry, it's more about owning the idea, pumping in a lot of money, investing time, resources, and the entire wherewithal available to ensure that this collaboration ends in a product. So most important thing is creating an ideal environment. That is one of the key things where there has to be transparency, there has to be trust in terms of the partners coming together to tackle a problem. For example, what we have seen in the last couple of decades is an unparalleled, unprecedented amount of ecosystem capacity building, wherein companies and academic labs have come together and have really built a know-how, and the fruits of which all of us are benefited in terms of a vaccine. In unprecedented speed, we had new drugs, new monoclonal antibodies, as well as vaccines which were developed. And how the whole thing was done is something which is kind of built around a lot of trust and also built around strong science. And what is very important in another collaboration is to define clarity on roles and objectives so that it's a win-win for both the partners. And the most important sticky issue, which I've faced in the last 20 years, how do you work around and have clarity on confidentiality and IP discussions? All these things can be done if the partners have an open discussion. Okay. If I move on to the next slide. So what I'm going to do in the next 30 minutes is to talk to you about Achila disease, malaria. As we speak, by the time I finish this talk for 45 minutes, we would have lost around 25 children because of malaria. Every 2 minutes in the world, a child dies because of malaria. And is malaria treatable? The answer is yes. Why is it that we're still not able to do it? Because it's a very complex biology of the parasite. There is drug resistance, which is coming out, and plus accessibility to medicines. So what I'm going to talk to you about is the journey of discovery of a novel anti-malarial molecule, which is called AZ. This was done at AstraZeneca, 13721412. Then subsequently, the product was developed by MMV, Medicines for Malaria Venture, with the code name 253, and now it is being developed by Zydas Cadila. So I will walk you through a wonderful, fantastic partnership between AstraZeneca, Medicines for Malaria Venture, and Zydas Cadila. A quick word about malaria. Malaria continues to be a huge burden on the society. It's a huge unmet need. What you're seeing here is a WHO report on malaria published in 2020, where the numbers are really depressing. 3.2 billion people across the world are at risk of acquiring malaria. And if you really put this into numbers, in 2019, 229 million cases of malaria were reported across the world, of which there were 400,000 deaths. And unfortunately, there is an unbalanced or a heavy toll on African continent. So 67% or 2/3 of the deaths happen in children below 5 years. As I put it in numbers, every 2 minutes a child dies. And the economic cost of malaria disease burden is close to $12 billion per year. And what is the world doing about it? There has been a huge uprising in terms of how do we control malaria. And another disease called tuberculosis, which probably in another occasion I would love to talk about. So there has been a lot of funding from philanthropists, from government agencies, to the tune of about $3 billion. This is a trickle in the ocean, but at least it's this investment is what is taking us forward. I will show you some phenomenal progress that has been made in the field of malaria prevention and treatment. So the major aim of tackling malaria is to cure malaria. We also have to, the parasite is getting smarter week by week, year after year. How do we tackle resistance? We do have a cocktail of drugs. I'll show you a slide, but still the parasite is developing resistance to the frontline drugs. And most importantly, how can you block transmission? And the interesting part is the onset of the COVID pandemic has had a huge impact on the ongoing success in terms of malaria control and treatment. So an estimate showed that a 10% disruption in effective access to anti-malarial drugs in sub-Saharan Africa could lead to an additional 20,000, 19,000 deaths. If the same reduction in access is 50%, which I think is a reality, because what has happened is the world has really responded in a big way to COVID, and in the process, all the other ongoing challenges in terms of disease burden have all been neglected. And I could confidently say that there's almost a 50% reduction in accessibility to malaria treatments, which is going to cause another 100,000 deaths each year because of malaria. So this story here this is a publication called triaminopyrimidine. It's a novel chemotype. It's a fast killer. It also has a long-acting anti-malarial clinical candidate, which was developed as part of this partnership. The work started way back in 2011, 2012, and we are talking in 2021 now. It's taken 9 years for where the molecule is. And based on an extensive survey done by McKinsey since 2000, the average time it takes for a molecule from the start of phase 1 till it's approved in the market is almost 10 years. And if you add the discovery time, which can take anywhere from 3 to 5 years, you're looking at a timeline of 20 to 15 years. Drug discovery is very punishing. The failure rates are very high. We'll talk about it as we go. It's brutal in terms of the amount of money that's needed and the time it takes to really develop a successful drug, which can bring benefit to patients. So the story here, who are the partners here? As I told you, the industrial partner, the work started at AstraZeneca. Subsequently, we had partnership with GlaxoSmithKline, as well as Zydas Cadila, which came into the picture later on. From academia, we had Icidev. Icidev is an institute where they have a screening facility to screen almost 500,000 to 1 million compounds against the asexual stage of Plasmodium falciparum. Then we work very closely with academic groups in Harvard and Columbia University to really understand the mechanism of action of this molecule. We are ably supported by government agencies, National Institute for Malaria Research in Delhi, as well as CCAP in Bangalore, in terms of access to high-end flow cytometry. And then we also had MMV, which stands for Medicines for Malaria Venture, a Swiss-based not-for-profit agency, which is really spearheading the global response to malaria. So a quick idea about Plasmodium falciparum. I wish this talk was delivered in live, where we could interact with students, ask questions and see the response and make it very interactive. Unfortunately, the platform does not allow this, but I will reserve some time for questions. So please make note of your points, and I really would be very happy if there are questions at the end of my talk. So Plasmodium falciparum, as all of us know, causes malaria. There are 4 major species. Plasmodium falciparum is responsible for almost 80-90% of malaria worldwide. Then you have Plasmodium vivax, which causes a form of malaria, which causes a lot of sickness. People don't die, but it causes a lot of morbidity. Then you have Plasmodium ovale and Plasmodium malaria. So malaria is very unique that you need an intermediate host. The intermediate host here is a mosquito, the female Anopheles mosquito. There are over 400 species across the world which can transmit malaria, depending on the geography, the weather, the host, and everything. There's a set of factors which contribute to this. So infection malaria starts when a mosquito bites an individual, a susceptible individual, and during the blood meal, the mosquito pushes in a lot of sporozoites. And the sporozoites are the one which are taken up. They go in, go to the liver, go through a cycle, and then they go through an extraordinary complicated life cycle. Why is it extraordinary? Because it's so beautiful that the parasite has really adapted well. And then it goes through different stages. So every 48 hours, there is a release of merozoites into the bloodstream. So that primarily coincides with the fever. So someone who has got malaria, I'm sure a lot of people in this audience here have had malaria at some time in their lifetime. So every 48 hours, there's a cyclical fever. That is because of merozoite release. And this cycle continues for many, just use my next pointer, continues for many cycles. And one fine day, the parasite decides to undergo sexual differentiation into male and female. That is taken up by the mosquito when it takes a blood meal from an individual who has malaria. And then it goes into the mosquito, and there it goes through a life cycle. It takes almost 14 to 18 days in the mosquito when the parasite has evolved into sporozoites. And this mosquito, when it bites another human, transmits malaria. So in terms of, so this is a cyclical representation of how much time does it take for the entire life cycle. The most important thing to notice, every 48 hours, when the merozoite is released into the bloodstream, you have clinical symptoms. And the intensity of transmission across the globe depends on the availability of the vector, the prevalence of the vector, the status of the biology of the parasite, whether it's a drug-sensitive parasite or a drug-resistant parasite, the susceptibility of the human host, plus the environment. The environment here is, do you have stagnating water? If it's very, there's a lot of rain, Southeast Asia, with the amount of rain we get is a perfect breeding ground for the parasite. So a quick cycle into the drug discovery process. All of you have heard, right? You take medicines for headache. Someone in the family takes a medicine for diabetes or hypertension. It takes 10 to 12 years. And the average industry estimate says it can cost anywhere from $1.8 billion to up to $3 billion, depending on how successful the models are available, how translatable the models are between the in vitro studies and the studies in humans. All put together, it's a very expensive endeavor. So we started millions of compounds. This particular project, we resorted to a cell-based screen, wherein we took the asexual stage of Plasmodium falciparum, which grows in culture, fed with O-positive blood with RBCs. On that, we screened 500,000 compounds from the AstraZeneca corporate library. These are molecules which have been built over decades, very diverse in terms of chemical space. The chemists out there will understand what I mean by diversity. So it's a cell-based screen based on which you look for growth inhibition of the parasite, and then we started with 500,000. then we ended up with close to 5,000 to 8,000 molecules, which showed growth inhibition in culture. And that went through a very elaborate, that's hundreds of molecules, went through what is called a lead evaluation. This is to look at what you get as a chemical to a drug is a long process. As I've been telling, it takes anywhere from 3 to 5 years to do a lot of chemistry to bring in, to dial in ideal properties and dial out unwanted properties. So this process is an iterative process, and then it goes through a set of assays. That's where biochemistry plays a very important role, microbiology plays a very important role. The entire palette of biological assays required to probe the different stages of the life cycle of the parasite becomes very important. And then we go through an iterative cycle. In this project, we made around 800 plus molecules over a period of 3 years, tested in hundreds of different panel of assays, finally to come up with a molecule. And that one molecule went for preclinical development, and then I will talk about where we are now. Am I still audible and I'm still connected? If someone can confirm, please.
Yes, sir.
Yes.
Sir.
Thank you. Yeah. So this is a moving target, right? So malaria, as I told you, is a disease which impacts the developing world. So the treatment has to be affordable. It has to be extraordinarily safe. It has to be something which can be given free of cost, right? It has to be compatible with other drugs, because chronic diseases like tuberculosis or malaria require a combination of drugs, because doing monotherapy is a recipe for disaster, because monotherapy induces development of resistance. So what we envisage is a mixture of drugs with different mechanisms of action, something targeting the DNA biosynthesis, something targeting the heme synthesis, something targeting protein synthesis. So to name a few. Ideally, what you're doing is you are basically trying to immobilize or inactivate the parasite by hitting it with different targets. So the most important thing to ask this question as we embark on the journey of drug discovery is why? Why control malaria? Control we want to eradicate, because malaria in decades just meant you want to get. So there are
Participants, kindly mute your mic.
Important concepts called it. There are two questions to ask. One is called the TCP, which is the target candidate profile. Like when we interview, let's say, so you come up with a set of questions, right? So similarly here in drug discovery, TCP3 could be anything which targets hypnozoites, which sit in the liver and keep causing
Sir.
Yes. Yeah. Can you hear me now?
Yes, sir.
Yeah, I got kicked out for a few minutes. I don't know what happened. Yeah. So I was talking about TCP. So we need to know which part of the life cycle of the parasite do you want to really target. So that is TCP. Then comes TPP, which is the target product profile. Is it a orally developed drug, delivered drug? Is it going to be given intramuscularly? Is it going to be given intravenously? And what will be the cost of monotherapy, or are you going to give it as combination? So it's a very challenging task to really to make sure that this is how you proceed. And then comes the question of how do you make this? It has to be cheap, as I told you, readily available, freely available to the people who need this. This is in the developing world. So So here is a drug discovery which has been put forth by Medicines for Malaria venture. Does the compound have activity against malaria? So does it have activity on the parasitic life cycle? Does it hit the asexual stage? Does it hit the merozoites? Does it block the differentiation into gametocytes? Then does it have drug-like properties? Is the chemistry novel? Because the industry is putting in a huge amount of investment to the tune of $1 billion to $2 billion. You want to know that there is clearly a space in terms of an IP protection, and also ask the question: does the mechanism of action be very novel, right? And then here comes called the make this cycle. As I told you, it's a iterative process which is for years together. Every day a new molecule has a new feature, a new property. And then the chemists, biologists, pharmacologists sit together to really understand how do you go about optimizing the molecule. So you do a hypothesis, you synthesize the molecule, you test them in a variety of biological assays, and then with the results you interpret and then come back. As I told you, over the last 6 years, Medicines for Malaria venture in terms of partnership has developed around 12 new candidates. So here is where the partnership really matters. For a disease like malaria, where there is no commercial potential, and the internet or target population is people living in the developing world, you really need to make, you just cannot go and build new assays, new capacity in each and every place. So the key is to really open up and build global partnerships and capacity building, which will really help partners to access. So what we're looking at here is a few of the assays I've shown here. This is probably around 8 to 10 very critical assays, which involves in vitro studies, which involves letting the mosquitoes feed on the molecule as well as on blood. Then you use alternate surrogate parasites to really understand the biology. Then we use mice, we use rats, we use macaques, non-human primates. Then we go through this entire cycle of really understanding how your molecule works. And we are very lucky to have this accessed by our partnerships built with academic collaborators as well as with industrial partners. So coming to the molecule, which is MMB253, as we spoke about target product profile, the target product profile, which was defined at the beginning of the project in 2011-12 was, it has to be a novel molecule, a novel chemotype, a novel mechanism of action, and it should work across all known isolates of Plasmodium falciparum, the existing ones as well as the new drugs that are being developed, resistant strains. It has to be equipotent against Plasmodium falciparum strains with known mechanism of resistance, so that it includes both the ones available in the market as well as ones which are in clinical development. It has to have a rapid onset of kill. Why is this important? As we spoke about, every 48 hours when the parasite is released in the bloodstream, you're looking at 10 power 8 to 10 power 10 parasites per ml of blood. That's a lot of parasites. So the molecule has to immediately kill these parasites to decrease the parasitemia so that there is no fever and a clinical response. The compound should have a long T-half. You're talking about people who live in remote areas in Africa or in India who cannot keep coming to the clinic every day to take the medicine. So you want to bring them to the clinic, test them, diagnose, give them a regimen of multiple drugs, and then they go off, that the disease is cured. It has to have good selectivity. Malarial parasite is a eukaryote, so it shares a lot of kinases and a lot of other targets with humans. So you really have to make sure that there are no side effects or tolerability issues coming because of cross-reactivity with kinases. The cost of goods have to be low to moderate. Literally, this is a killer, right? The entire process is expensive. It has to be dirt cheap. And where are we, right? As I mentioned, we ran 500,000 compounds against actively growing Plasmodium falciparum in a Petri dish, a 1536 well Petri dish. And then the initial molecule which we picked up, which is called a hit, had a IC50 inhibition concentration 50%, nanomolar of 670, which is roughly 0.6 micromolar IC50, which a lot of people in academics will jump and say, wow, this looks great, but that is really not enough. Over the period of 3 years of optimization, we developed a candidate where the potency dropped to 10 nanomolar, that is 0.01 micromolar, 0.001 micromolar. And then we also had to ensure that, as I told you, this particular molecule had a good degree of activity against certain kinases, especially the acetylcholinesterase receptor, which is present in the cardiac muscle and other parts of the human body. So original window of differentiation or the therapeutic window was only 0.05. During the optimization process, it increased to a window of 500-fold. Then it also had activity against the hub channel, which controls the activity of the cardiac muscle. So the IC50 to Cmax ratio was 17-fold. We increased it to 230-fold. So basically, this entire exercise, I'm cutting short 4 years of work into one slide to show you the kind of optimization that has happened. So for those of you who love chemistry, here is a molecule. It's a triaminopyrimidine. The original molecule had 150 nanomolar IC50. Solubility was around 10 micromolar. Okay. It had good activity against the hug channel, so which was what we had to really overcome. Over the period of different multiple rounds of optimization, you could see that the IC50 went to 600 nanomolar, solubility increased, other things reduced. Finally, when we landed up with our preclinical candidate, why is it called preclinical? Is because once you narrow down to a candidate, then you take it through an elaborate and very expensive endeavor to understand, is this molecule going to stand the test of time in terms of safety, tolerability, in at least 2 animal species to begin with? And will all the results you see in these 2 animal species translate into safety humans when we phase 1, phase 2, phase 3 trials? As you could see, the preclinical candidate, the IC50 dropped down to 9 to 10 nanomolar. The hug IC50 increased to 33 micromolar. And then we had a molecule which was soluble. And it also had a low activity or a good degree of selectivity across other kinases. So in the TPP, I showed you that the molecule what we are going to work on should be a rapid killer. So this is an in vitro assay where we basically grow parasites. As you could see that we have around 1 million parasites per ml in a well, in a microtiter plate. And for comparison, so this is the molecule which we call compound 12 at the time. You can see the one with the inverted triangle. You could see that within 24 to 30 hours, we see a 6-log reduction in the parasite number. As a comparison, I put chloroquine, which has been the standard anti-malarial drug, which has really helped to control malaria to a large extent across the world for many, many decades. So this you could see also kills rapidly, and our molecule is slightly better than that, or it basically benchmarks with chloroquine. Then we have another molecule called pyrimethamine, which is also used across the world. You could see pyrimethamine is a slow action. It does not kill as rapidly as our molecule. Another new molecule called otavacone, which is used as a prophylactic treatment. So here again, you can see that this is a much of a slow killer. So this is basically to benchmark where do we stand in the spectrum of different molecules. Then most important thing is, so this drug what we developed has to be given in an oral formulation, basically a tablet. So we need to ask the question, every time I give a drug, how is it absorbed? I'm sure many of you have heard about this term called pharmacokinetics, which is basically body. When you talk about human body, we're talking. So pharmacokinetics is basically following order of what we're doing is, as you keep increasing the dose, there increase in the great news because not yet reaching saturation. And hello, sorry, got kicked out. I don't know where the technical problem is.
It's okay, sir.
Am I still audible?
Yes, sir. Yes, sir.
I'm just going to share my screen again. Yeah. Can you see the screen, please? Can someone confirm? Yeah.
Yes,
Okay,
sir.
Maria, can you
Yes, sir.
confirm?
It's visible.
Yeah, sure. So the graph on the left side talks about a blood concentration was the time profile. The good news here is the molecule is not getting saturated in a short span of time. So we're able to really increase the dose. And as we measure the drug level, we also do an in vivo model in a mouse model of malaria, wherein we use what is called a humanized mouse model, because Plasmodium falciparum does not grow in mice. So you really have to condition the mouse or the mice to remove their blood and then infuse with human blood. So it's called the humanized mouse model. Very expensive, extremely fragile system in which we're able to measure in the absence of drug, the parasite grows. When we give increased dose of the molecule, we're able to kill and then take it to LLOQ, which is lower limit of quantitation. Then came the big question. The other participants, could you please mute once you are not talking?
Participants, kindly mute your mic.
Thank you. So as I told you, so this molecule came out of a cell-based cream, right, of against Plasmodium falciparum. So we did not know what it targets. We see that as you increase the dose, it kills the parasite in culture, in petri dishes. As you increase the concentration of the drug in animal models, in a humanized mouse model of falciparum, it kills and cures malaria or it reduces the parasitemia. So we didn't know what the target was. So, as biochemists, you all must be aware of doing a genetic selection. So basically, we grow the parasite in the presence of increasing concentration of the drug, and then ask the question, can I get a parasite which will grow in the presence of drug? So such a thing is those parasites which develop resistance in culture over a period of time are isolated, cloned, and then we do a whole genome sequencing and identify the target. At the time when the paper was published in 2015, we found that because of collaboration, we could find that the resistance to triimmunopyrimidines mapped to the vacuolar ATP synthase. The parasite sits inside an RBC, inside a vacuole, and it requires a huge amount of energy to really transport its nutrients. So this molecule happens to hit something on the vacuolar ATP synthase, cause a point mutation, because of which the parasite is not able to take its nutrients and then results in parasitic death. So we are not sure whether it's a mechanism of resistance or mechanism of action. So this work is still ongoing. I believe that this year we should have a publication from a collaborator where they show that they have identified heme biosynthesis as the target for this molecule. Then comes the most important aspect. Any drug that goes for human use has to pass through a battery of tests in different animal species, non-human primates, to show that it is safe. So all I want to show in this slide here is on the left side is when we had the first molecule, the selectivity of 100-fold, which is required to safely dose humans to reduce the burden of malaria, was not achieved. Whereas by the time we came to the final molecule, the clinical candidate, over a period of 4 years, we could get at least 100-fold selectivity across different receptors, meaning to say that at the dose, intended human dose, we still did not inhibit the receptors what are shown here. So there is a panel of around 100 different kinases representing the entire human body, which is frequently tested, assessed to make sure that there is actually no activity or very less activity. So MME253, the final molecule, this is where we are. Mechanism of action at that point in time was it probably hits the ATP4, the vacuolar ATP synthase. This is the molecule. Those of you who understand chemistry will know this looks beautiful, right? So the IC50 was around 10 nanomolar. It works. It did not show any activity on the liver stage. It killed the gametocytes, which means that it can block transmission. Then this parameters in the middle shown here in yellow are basically in terms of how does the molecule get metabolized. And bioavailability is an important parameter where we show once you take a tablet via the oral route, how much of it is available in the systemic circulation. The half-life of the molecule was around 14 to 16 hours at the time when we estimated. And the effective dose 90 is around 13 milligrams per kilogram. Okay, so then everything was going well. We are poised to develop. Then in 2014, AstraZeneca, because of a global strategic re-initiative, said that they're going to shut the R&D center. So the R&D center where all the work happened in Bangalore, in Hebal, was closed by May 2014. So all of us lost a job. Then we really had to find a partner to give the molecule back. Fortunately, at that point in time, MME253 was ready to go into phase 1 or into preclinical development. Subsequently, to kilogram to really reduce costs, all of this has been brought down. Plus, also embark on a phase 1 trial. Long story short, has again, I got kicked out. Is my screen visible?
Not it, sir.
Is it visible
Okay,
now?
sir. Yeah, yes, sir.
Yeah. So the molecule now called ZY19489 was tested in human volunteers in Australia in an increasing dose all the way from 50 to 100 milligrams all the way up to 1,500 milligrams. It was well tolerated. The half-life, interestingly, in humans, in mouse, we saw it was 30 to 14 hours. And based on all the predictions, we knew that the half-life in humans would be higher. But we got the actual data showing that it has a half-life of around 80 hours, which is fantastic. The parasite kill rate was comparable with artemisinin, which is the frontline anti-malarial drug. The PKPD modeling, which is pharmacokinetic pharmacodynamic modeling, showed that a dose anywhere between 450 milligram to 1,000 milligram should result in 6 log or 9 log parasite killing. And the data from the study showed that this molecule is ready to progress into phase 2. Quickly, I want to introduce to you another concept. The biggest challenge in drug discovery, why drugs fail, is what you do in test tube, in vitro culture, what you do in animals does not translate into humans. So that's the value of that. A lot of molecules, it looks fantastic in culture in all the in vitro assays, but the moment you move to humans, it does not work. In malaria, there's an interesting model called the volunteer infection studies. Basically, what you do is you take a known drug-sensitive parasite, Plasmodium falciparum, inject it into volunteers. It could be college students or it could be any healthy volunteer, and let the parasite develop. And you know that if the parasite goes out of control, you can always treat it with a cocktail of drugs. So after 14 days, after 7 days, you administer the candidate drug. In this case, we administered MME253, and we could measure whether the drug actually gets absorbed into the system after being delivered orally. Does it reach adequate concentration in human volunteers? And does it also kill the parasite? So we get a fantastic readout in terms of both exposure, which is pharmacokinetics, as well as pharmacodynamics, which is whether the drug kills the parasite in a human volunteer. So we are very lucky to have this VIS study model developed by Medicines for Malaria Venture. And this model was utilized and then shown that we could actually see how the drug works in human volunteers. And this model is really going to help in terms of identifying and finding the right partner drug. So overall, the phase 1 study from Icidev Cadila in partnership with MMB, this is the overall outcome. The phase 1 healthy volunteer study demonstrated long half-life of around 80 hours. And the drug has a potential for a single dose cure. If you remember, I showed you in the TCP and TPP that we want a drug which has to be administered only once. Because imagine a child living in Africa. You cannot expect a child living in a remote area to come back to the clinic every day to take drugs. So it has to be a single dose in a cocktail. And this drug actually meets those criteria. And it also shows excellent efficacy or a good parasite killing in a human volunteer model, which is really remarkable. And it's fast acting, and it also works on falciparum and vivax across the world in terms of all the new strains. So where are we now? So this is the portfolio of Medicines for Malaria Venture. It's a rich portfolio over in the last 20 years, Medicines for Malaria Venture, because of their ability to stitch partnerships with industry and academia across the world, and find mechanisms to continue to fund and identify the best projects which should go forward. And in the process, have a very robust decision-making tree, where only molecules which have chances of success go forward. The ones which you know is going to fail in 1 year should be killed today. So in drug discovery, they say that fail early, fail safe, fail cheap, which means that if a project dies in the screening phase, you save a lot of money and time. If a project fails in phase 1 or phase 2, you already would have spent close to $500,000, $500,000,000. So it's very important that we have decision trees in terms of which one goes forward. So happy to say that the molecule has finished phase 1 and is waiting to do a combination with the other partners that are in development into a phase 2 study in malaria patients. Then if found safe, tolerated, and efficacious, will go into a global phase 3. We're looking at another 5 to 6 years before the drug can successfully finish a phase 3 and then be available in the market. So in summary, triimmunopyrimidine, which is now called ZY19489, has the potential to be a single dose cure for Plasmodium falciparum and Plasmodium vivax malaria. This molecule demonstrates a novel mechanism of action with a long half-life of around 80 hours in humans. It exhibits rapid parasite killing. What we saw in vitro in a culture translated in humanized model of malaria in mice. It also translated in the VIS model of human volunteers. And it kills all the intra-erythrocytic malarial stages, as we spoke about. The sporocytes go in, then your merozoites, then the different cysions, all the different phases. It looks like it has activity on those stages. And most interestingly, so far, we've not been able to raise a resistant mutant, which shows that the propensity to develop resistance to this molecule is extremely low. So I want to leave this slide with a lot of hope. As I told you, MMV and a lot of other global agencies, including WHO as well as Indian health agencies, have been working towards bringing in the best minds, best technology, best know-how to really address this problem of malaria. What you're seeing here is taken from the World Malaria Report of 2020. From 2000 till 2020, you could see that there has been a steady decline in the number of malaria cases as quantitated by number of cases per 1,000 population at risk. So this is the same cohort of people living in Southeast Asia and Africa, where we're looking at the incidence. As you could see that in 2000, the incidence was around 80 cases per 1,000 population, whereas in 2020, it's 56.8. There is almost a 40% reduction in the incidence, which is good news. And most important is mortality, right? One is getting the disease. If you live, it's great. If you die, it's really unfortunate, right? So mortality rates have significantly dropped from 25 per 100,000 population to 10 per 100,000. And the bar is going down. Let's hope and pray that the COVID-induced disruption in availability of antimalarial drugs in terms of available diagnostics is not really going to move this bar up. That was on the right is Dr. Shaul Hamid. He is a very creative. The colleague you see on the left side is Dr. Suresh Olapare, who is them out of brain tumor. I really would like to dedicate this talk to my listening. I'm sorry that disruption, I think twice of the issue. Otherwise, I'm glad to take any. You have to be very happy.
Sir, am I audible?
Participants, kindly post your questions in the chat box. Our guest will be answering your questions once he joins.